ನಮಸ್ಕಾರ ಪ್ರೀತಿಯ ವಿದ್ಯಾರ್ಥಿಗಳೇ. ಕರ್ನಾಟಕ ಸರ್ಕಾರ ಪದವಿ ಪೂರ್ವ ಶಿಕ್ಷಣ ಇಲಾಖೆಯಿಂದ ನಡೆಸುತ್ತಿರುವ ಜ್ಞಾನ ತರಂಗ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ತಮಗೆಲ್ಲ ಸ್ವಾಗತ. ಈ ಕಾರ್ಯಕ್ರಮದ ಮೂಲಕ ತಾವು ಸ್ಪರ್ಧಾತ್ಮಕ ಪರೀಕ್ಷೆಗಳಿಗೆ ತಯಾರಾಗುತ್ತೀರಿ ಎನ್ನುತ್ತಾ ನಾನು ಡಾಕ್ಟರ್ ಎನ್ ಬಗಾಡೇಕರ್ ಇಂದು ಪ್ರಥಮ PUCಯ 3 ಪಾಠಗಳನ್ನು ನಿಮ್ಮೊಂದಿಗೆ ಡಿಸ್ಕಷನ್ ಮಾಡುತ್ತೇನೆ. ನಾನು ಮಾಡುತ್ತಿರುವ 3 ಪಾಠಗಳು, the first one is from Unit 1, that is Plant Kingdom. Then from Unit 4, that is Plant Physiology. Chapter 11, that is Transport in Plants. And Chapter 15, Plant Growth and Development. Let us start with Plant Kingdom. ಇದರ ಒಂದು ಸಣ್ಣ ಪಕ್ಷಿ ನೋಟ ಸಿನಾಪ್ಸಿಸ್ ರೂಪದಲ್ಲಿ ನೋಡಿಕೊಂಡು ನಾವು ಕ್ವಶನ್ ಆನ್ಸರ್ ಅನ್ನು ಡಿಸ್ಕಷನ್ ಮಾಡೋಣ. ದಟ್ ಈಸ್ ಟಾಪಿಕ್ 3, ಪ್ಲಾಂಟ್ ಕಿಂಗ್ಡಮ್. ಈ ಆರ್ಗ್ಯಾನಿಸಮ್ಗಳನ್ನು ಕ್ಲಾಸಿಫೈ ಮಾಡೋದಕ್ಕಾಗಿ ಲೀನಿಯಸ್ ಅವರು ಆರ್ಟಿಫಿಷಿಯಲ್ ಸಿಸ್ಟಮ್ ಆಫ್ ಕ್ಲಾಸಿಫಿಕೇಶನ್ ಅಂತ ಮಾಡಿದರು. ದೇ ಹ್ಯಾವ್ ಕನ್ಸಿಡರ್ಡ್ ವೆರಿ ಫ್ಯೂ ಅಬ್ಸರ್ವೇಬಲ್ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಟು ಕ್ಲಾಸಿಫೈ. That is called as artificial system of classification. Then Bentham and Hooker classify these organisms, especially flowering plants, into observable characters and internal characters. That is, internal character and external characters both are considered for that natural system of classification. Then this phylogenetic system of classification includes evolutionary relation among the organisms. Next comes numerical taxonomy. This includes nowadays bioinformatics, that is in biology computers are used. Using those computers, if we are classifying the organisms by including all the datas, then that is called as numerical taxonomy. Then comes cytotaxonomy. In this cytotaxonomy, we will consider chromosomes for the classification. Structure and behavior will be considered in cytotaxonomy. Next comes chemotaxonomy. In this chemotaxonomy, the chemical constituents present in the cell or organisms are considered for the classification. So, we all know that R.H. Whittaker in the previous classes classified the living organisms into 5 kingdoms. That is kingdom Monera, Protista, Mycota, Plantae and Animalia. Okay, and today we are discussing with this, that is kingdom Plantae. And in kingdom Plantae again they are classified into 5 different groups, that is algae, bryophytes, pteridophytes, gymnosperms and angiosperms. So let us start with algae. Algae are chlorophyll bearing, simple thyloid, autotrophic and largely aquatic organisms. Means ಇವು ಮುಖ್ಯವಾಗಿ ನೀರಿನಲ್ಲಿ ಕಂಡುಬರುವಂತಹ ಗಿಡಗಳು ಮತ್ತು ಇವುಗಳ ಶೇಪ್ ಥೈಲಾಯ್ಡ್, ಅಂದರೆ ಎಲ್ಲಿ ಗಿಡಗಳನ್ನು ನಾವು ಎಲೆ, ಕಾಯಿ, ಬೇರು ಅಂತ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಮಾಡ್ಲಿಕ್ಕೆ ಬರಲ್ಲ ಅಂತಹ ಗಿಡಗಳಿಗೆ ನಾವು ಥೈಲಾಯ್ಡ್ ಪ್ಲಾಂಟ್ಸ್ ಅಂತ ಹೇಳ್ತೇವೆ. ಅಟೋಟ್ರೋಫಿಕ್ ಮೀನ್ಸ್ ದೇ ಆರ್ ಫೋಟೋಸಿಂಥೆಟಿಕ್ ಆ್ಯಂಡ್ ಹಿಯರ್ ದ ಫಾರ್ಮ್ ಆ್ಯಂಡ್ ಸೈಜ್ ವೇರೀಸ್. ಅವು ಬೇರೆ ಬೇರೆ ಅವುಗಳ ಸೈಜ್ ಮತ್ತು ಶೇಪ್ ತುಂಬ ವೆರೈಟಿಯಲ್ಲಿ ಇರುತ್ತೆ. ರೀಪ್ರೊಡಕ್ಷನ್, ದೇ ರಿಪ್ರೊಡ್ಯೂಸ್ ಬೈ ವೆಜಿಟೇಟಿವ್, ದಟ್ ಈಸ್ ಬೈ ಫ್ರಾಗ್ಮೆಂಟೇಷನ್. Asexual reproduction mainly through zoospores, and sexual reproduction they reproduce mainly by 3 types, that is isogamous, anisogamous and oogamous. Next, how these algae are important? They are very much important in carbon dioxide fixation. Almost 50% of carbon dioxide is fixed by these algae. Then these are the primary producers for many aquatic animals and they are also used as food. And these algae are also having commercial importance. They are hydrocolloids and they are commercially used, and the hydrocolloids are in the form of algin and carrageen. And agar is used in laboratories, in tissue culture laboratories and microbiology laboratories. For culturing they are using this agar, and also ice cream and jellies they are used this agar. Food supplements by space travelers means chlorella and spirulina, they are very rich in protein. That's why these are used as a food supplements. Then we can see here various forms, and also based on their pigments present in that algae, they are classified mainly into three types, that is chlorophysae, PHYSIOLOGY and rhodophysae. Chlorophysae is having pigment called as chlorophyll A and B. That's why these are having this green color. The major color is green and they store food in the form of starch. Whereas PHYSIOLOGY is brown color, olive green to brown color based on the how much fucoxanthin present in it. It may be olive green to different shades of brown colors present in this PHYSIOLOGY, and these are commonly called as brown algae. Okay, and in rhodophysae they are having red pigment called as R-picoerythrin. That's why these rhodophysae are also appear red in color, that's commonly called as red algae. So algae are mainly classified into three types: chlorophysae, PHYSIOLOGY and rhodophysae based on pigments present in it. Next we will move for bryophytes. Bryophytes are called as amphibians of plant kingdom. Thallus is erect or prostrate, means thallus is erect like this or prostrate in mosses. In mosses it is prostrate body. They lack root, stem, leaves. There are no true roots, true stem or leaves. Here in gametophyte is a dominant phase, and that gametophyte will produce antheridium. Here in antheridium it produces antheridium, and here in archegoniospore, which is with archegonia, a female representative and it is a male representative. And importance, these in bryophytes dominant phase is gametophytic phase where it produces antheridium and archegonium. In this thallus it will produce antheridium, which is having male reproductive gametes, that is antheridium with antherozoites, and here archegonium is a female reproductive part with archegonia. And here it is we are classifying it into mosses and ferns. Then importance, it is used as a food for very few animals, and mainly that sphagnum, because it is having a water holding capacity, it act as a peat. Next we will move for pteridophytes. Pteridophytes are the first vascular plants, means these are the plants with having a xylem and phloem for water conduction and food conduction. Not only that, here we are not using the term thalloid form in plants. Here we can use it is complete organ system, means it is having roots, stem and leaves. Here we can very easily get the true roots, true stem and leaves. In this, sporophyte is dominant phase, and it is also having very short lived gametophyte which is free living photosynthetic, and that gametophyte is called as prothallus. It bears antheridium and archegonium. Sporophyte is a homosporous or heterosporous. As name itself, homo means similar, means spore size are similar, or heterospores means they varies in their size, and we can represent the larger one as a female and smaller ones as a males. And here these pteridophytes are classified into four classes, that is Psilopsida, Lycopsida, Spinopsida and Pteropsida. Next we will move for gymnosperms, that is they are naked seeded plants. Naked seeded plants means ovules are not enclosed by any ovary wall and remain exposed. And here roots are associated with the mycorrhiza in Pinus, cyanobacteria in corollary roots of cycas. Here stem may be branched or unbranched. Unbranched ಅಂದರೆ ನೀವೆಲ್ಲರೂ coconut tree ನೋಡಿರ್ತೀರಿ, ಅಲ್ಲಿ stem unbranched ಇರುತ್ತೆ. ಅದೇ ಥರ ಇಲ್ಲಿ ಕೂಡ stem unbranched ಆಗಿರಬಹುದು, ಅಥವಾ ಕೆಲವು ಸ್ಪೀಷೀಸ್ಗಳಲ್ಲಿ ಅದು branched ಆಗಿ ಇರುತ್ತೆ. Leaves are well adapted. And heterosporous, again here also they produce male cones and female cones. Then male strobili, that is male cone is formed. Within that male cone we will get clusters of microsporophylls, and those microsporophylls are with microsporangium. In that microsporangium microspores are produced. Like that, female strobili or female cone is made up of clusters of megasporophylls, and those megasporophylls are having megasporangium, and within that megasporangium the female reproductive gametes, that is megaspores, are produced. Next we will move for angiosperms. Pollen grains and ovules are developed in the structures called flower. ಹೂ ಬಿಡುವ ಸಸ್ಯಗಳು ಮತ್ತು ಹಣ್ಣು ಬಿಡುತ್ತವೆ, ಹಣ್ಣುಗಳಲ್ಲಿ ಬೀಜಗಳು ಕಾಣುತ್ತವೆ. Wide range of habitat and size means they have very wide range of habitats, like they can withstand with a very high cold with high temperatures, and they are varied in sizes also, means they are with microscopic wolfia to tall tree eucalyptus. They provide food, fodder, fuel and medicines. These angiosperms are mainly classified into two classes, that is dicotyledonnae and monocotyledonnae. Pollen grains are male gametes and ovules with embryo sac. Pollination takes place as stamens with the anther will produce pollen grains, which are the male gametes are dispersed through pollinating agents, and then that is landing on the stigma of the female part, and there is a pollination. After pollination there is a fertilization, and in that fertilization here peculiar double fertilization is taking place. You will study in detail in the next chapter. These are the dicots and monocot plants. You all see that jawar, grass, ragi, all monocots, dicots, all beans, legumes are dicots with 2 cotyledons, and whereas monocot is with a single cotyledon. Let us start with some of the examples or the questions to solve related to this topic. The first one is classification based on cytological information like chromosome number, structure, behavior is called. Whether it is a numerical taxonomy, cytotaxonomy, chemotaxonomy or natural system of classification. Classification based on cytological information. So, numerical taxonomy, as earlier we have discussed, it is based on bioinformatics, so this is not the right answer. Chemotaxonomy is related with its chemical constituents, so this is also not the correct answer. Natural system of classification, that is Bentham and Hooker's classification, where he considered external and internal characters, so this is also not the right one. So, the right answer here is cytotaxonomy. Next, which of the following pigment is present in the members of Rhodophysae? Rhodophysae means red algae. The color, red color, is because of which pigment is the question. Phycoerythrin is a pigment, and then chlorophyll B, then chlorophyll C and fucoxanthin. These are the 4 options. Chlorophyll B is present in Chlorophysae members, chlorophyll C is present in xanthophylls, and fucoxanthins are also present in brown algae. Chlorophyll C is also present in brown algae, B is present in chlorophyll B is present in Chlorophysae members. So, this is Rhodophysae question. So, Chlorophysae means this is not the right answer. Chlorophyll C is present in brown algae. So, Rhodophysae is a red algae and it is present in brown algae, so this is also a wrong option. Then fucoxanthin is a type of xanthophyll again present in brown algae but not in rhodophysa or red algae member, so this is also a wrong answer. So the right answer for this is R. phycoerythrine. We will move to the next question. Green colored multicellular asexual birds called gemmacups or gemmacups on which of the following organisms? Whether it is a Markensia, a Sphagnum, Funaria or Porphyra. Here you can see these three examples belongs to bryophytes and this is algae example. And if you recall your memories in the textbook, Sphagnum, there is a sporophyte is there, in Funaria sporophyte is there on that gametophyte, Porphyra is a algae, and in Markensia a thallophyte where these kind of asexual but specialized asexual beds are produced called as gemmacups are present on Markensia. So the right answer is Markensia. Just we can have a look on this. See, these are the asexual birds present within this cup and this cup is called as gemma cup. Next question is, coralloid roots are present in which of the following plants? Whether coralloid roots are present in pinus or they are present in legume roots, whether they are present in a rhizobium or cycas. ಇವು ನಾಲ್ಕರಲ್ಲಿ ಯಾವುದರಲ್ಲಿ coralloid ರೂಟ್ಗಳು ಇರುತ್ತವೆ? Pinus is an example for gymnosperm, cycas is an also example for gymnosperm, and here legume roots are there, rhizome are there. Here you can connect these two legume roots. Here there are no coralloid roots, okay. They are rhizobium which forms root nodules in this. Root nodules. Rhizobium is a bacteria which forms root nodules in legumes. So these two options are wrong. And in pinus, mycorrhiza is associated and they don't have any coralloid specialized roots here. So this is also a wrong answer. So the correct answer here is cycas with a coralloid roots. See, here we can see in cycas, these are the coralloid roots which are associated with cyanobacteria, and these cyanobacteria which are symbiotically associated with cycas roots help in nitrogen fixation. So the next question is, coralloid roots are present in which of the following? The options are pinus, legume roots, maize, cycas. Pinus, as we all know, it is associated with mycorrhizae, so this is not the answer. Legume roots are also associated with bacteria, but they are no. Legume roots are with root nodules which are associated with symbiotic bacteria called as rhizobium. So their root nodules are present but not the coralloid roots, so legume root is also a wrong answer. Maize roots, they don't have any such kind of coralloid roots, so the maize is also a wrong answer. But whereas in cycas, we can observe here coralloid roots. These are associated with cyanobacteria, and these cyanobacteria will give nitrogen to the cycas plant. In turn, it gives nutrition to those cyanobacteria, and these are symbiotically associated in cycas roots. So the coralloid roots are present in cycas. So the right answer is cycas. Next question is, Selaginella and Salvinia are considered to represent a significant step towards evolution of seed habit because embryo develops in female gametophyte which is retained on parent sporophyte. Female gametophyte is free and get dispersed like seeds. Here they are not dispersed like a seed and female gametophyte is not free, so this is not the right answer. Female gametophyte lack archegonia, again a wrong option. Megaspore possesses endosperm and embryo surrounded by seed coat is again here in pteridophytes is a wrong answer. So, the right answer here is embryo develops in female gametophyte which is retained on parent sporophyte. So, the right answer is A. Next question is, in bryophytes and pteridophytes, transport of male gametes require. For transferring the male gametes, what are the agents like? Here the first option is bird, second option is an insect, third option is wind and fourth option is water. Birds are not involved here and ornithophilia comes for pollen grain transfer or for fruits or seed transfers. So, here birds are not an option. Insects are also not involved, that is entomophilia comes under pollination. Wind is also not an option. Here pollen grains are transferred through wind and they are called as anemophily. But as because we know bryophytes and pteridophytes, here even though bryophyte is amphibian, they dependent on water for their reproduction. Pteridophytes are terrestrial plants, even then they require water for movement of antherozoids. So, both bryophytes and pteridophytes need a water for male gamete transport. So, the right answer here is water, that is D option is correct. Next we will move for question number 7, that is, which of the following statement is correct? The leaves in gymnosperms are well adapted to withstand extreme of temperature, humidity and wind. Water is not required for transfer of antherozoids of pteridophytes. Bryophytes are first terrestrial plants to possess vascular tissues. Algae are also called amphibians of plant kingdom. So, water is not required for the transfer of antherozoids of pteridophytes is a wrong answer. Just previous question, we know that water is required for transfer of antherozoids. Bryophytes are the first terrestrial plants. No, we know that first vascular plants are pteridophytes, not bryophytes. So, this statement is also incorrect. Algae are also called as amphibians of plant kingdom. Bryophytes are called as amphibians of plant kingdom but not algae. So, this is also a wrong option. The leaves in gymnosperms are well adapted to withstand extreme temperature, humidity and wind is a correct answer because the leaves present in angiosperms, they have modified, the stomata are sunken stomata and the leaf is with leaf surface is shiny and with thick cuticle. All those things they have adapted to check rate of transpiration. So, the right answer here is A. Next question is, in angiosperms, functional megaspores develop into whether it is a pollen sac or a embryo sac or nucellus or endosperm. Angiosperm functional megaspore is not developing into pollen sac because pollen sac is present in anther and which is a male representative of the flower. So, pollen sac is a wrong answer. Nucellus is a nutritive tissue. So, directly megaspore is not developing into nucellus tissue. So, nucellus option is also wrong. Endosperm formed after the fertilization, double fertilization and then triple fusion, that is PE, and primary endosperm nucleus forms endosperm. So, this is also a wrong answer. the remaining option is option B, that is embryo sac. Embryo sac is a right answer. Here monosporic type of development takes place where 3 megaspores degenerates and one of the megaspore developing into embryo sac. So the correct answer is embryo sac, that is option B. Next question is, free living photosynthetic thyroid gametophyte in pteridophytes is called, whether it is called as a thallus or prothallus, protonema or leafy stage. We all know thallus means undifferentiated plant body, where we cannot differentiate that plant body as a root, stem, leaf, flower, fruit. Such plant body is called as thallus. So this is not the correct answer. And if we study about developmental stages of mosses, there we can have 2 stages, that is protonema stage and second, this is primary protonema or protonema stage, and leafy stage or secondary protonema stage, which are related to bryophytes, not pteridophytes. So this is also a wrong answer. Prothallus is the right answer, where it is very short lived free living photosynthetic gametophyte present in pteridophytes. So the right option here is prothallus. Next question, you may get question like diagrams, unlabeled diagrams are there that we have to identify. Examine the figure given below and select the right option giving all the 4 parts A, B, C, D correctly identified. See, this is a NCERT textbook diagram where we have to label what is A, B, C and D. This is the example where you can see these are the air bladders. So this is B. A is a air bladder. B, leaf like structure called as frond. C, this is mid-rib. D is hold fast. This is frond and this is air bladder. So the right option here, it is not a first, it is not a frond, midrib, air bladder, holdplast. So this is not the correct answer. Holdplast, air bladder, frond, midrib is also not a correct answer. Midrib, holdplast, frond, air bladder is also not a correct option. The correct option is air bladder, frond, midrib, holdplast. This is an example of brown algae called as fucus, where these air bladder help it to float on the surface of the water. So the correct labelings of this diagram is option B, which is with air bladder, frond, midrib and holdplast. Next question is, microscopic smallest angiosperm is, whether it is a eucalyptus or whether it is a wolffia or whether it is a grass or whether it is a sequoia. Eucalyptus is exactly opposite to this question, that is the tallest angiosperm. So this is wrong answer. Grass, it is a small herb we can see through our naked eyes, not that small like we need a microscope. So grass is also not an correct example for smallest angiosperm. Sequoia is the tallest tree, belongs to gymnosperm. So sequoia is also a wrong option. So the remaining option is wolffia, which is a smallest angiosperm. sperm. We can see here in the fingertips we will find minute numerous plants. These are Wolffia species, and so the correct answer here is Wolffia. Next question is, select the wrong pair from the following. We have to select wrong pair among this. Just check here, bryophytes, Markensia and sphagnum. Pteridophytes, Selaginella and Xylotum. Phaeophysae, Laminaria and Sargassum. Rhodophysae, Volvox and Ulothrix. So, bryophyte examples, yes, here they have given examples. Bryophyte examples are Markensia and sphagnum. This is for Thallophyta and this is for Maasus. Examples are correct. So, this is a right pair. So, this is not a wrong pair, but for here it is a wrong option. Next is pteridophytes. Example, Selaginella and Xylotum. So, Selaginella and Xylotum belongs to pteridophytes. So, this is right match but wrong pair here. Phaeophysae, Laminaria and Sargassum. Laminaria and Sargassum are examples of brown algae. So, this option is also a wrong. Rhodophysae examples Volvox and Ulothrix. Rhodophysae members are Gracilaria or Polycyponia, Gelidium. Those are the examples but not Volvox and Ulothrix. Why? Because Volvox and Ulothrix are the examples of Chlorophysae, not Rhodophysae. So, the wrong pair here is option D is a wrong pair. So, we will move for the next question. Which among the following algae is a unicellular? And the following options are, option A is a Volvox, option B is a Ulothrix, option C is a Spirogyra and option D is a Chlamydomonas. See, Volvox is an example of algae, but which kind of organization here? It is colonial. Volvox is a colonial form, so it is not a unicellular. Ulothrix is a filamentous form. Ulothrix is filamentous form, so it is not also a correct option. Spirogyra is also filamentous form. So, this is also a correct, not a correct option. Whereas, Chlamydomonas is unicellular. Therefore, this is the right option. So, D, Chlamydomonas is right option for this question. Next question is, select the incorrect match. Select the incorrect match and the options are, option A is Pyrogyra, starch, Polycyphonia, Floridian starch, Volvox, Algin, Laminaria, Laminarin. So, if you observe Spirogyra is an example of Chlorophysae. In Chlorophysae member, reserve food material is in the form of starch. So, it is a correct match, it is not an incorrect match. So, this option is wrong. Polycyphonia is an example of red algae. Red algae is storing food in the form of Floridian starch. So, this is also a wrong option. Laminaria is a brown algae which stores its food in the form of complex carbohydrate, that may be a laminarin or mannitol. Here the option is laminarin is there. So, laminarin is also a right match, not an incorrect match. So, this is also not an option. Floridian starch is also not an option. Whereas, Volvox is a green Chlorophysae member algae which stores food in the form of starch but not in the form of algin. Algin is present in brown algae produced by cell wall but not in Volvox. So, this is hydrocolloidal substance used commercially that is not the correct option for Volvox algin. So, here incorrect match is algin C. Volvox algin is a incorrect match. ಇಲ್ಲಿ ಎರಡು ಆಪ್ಷನ್ ಕೊಟ್ಟಿದ್ದಾರೆ, ಸ್ಟೇಟ್ಮೆಂಟ್ ಎ ಮತ್ತು ಸ್ಟೇಟ್ಮೆಂಟ್ ಬಿ. ಸ್ಟೇಟ್ಮೆಂಟ್ ಎ ಏನು ಹೇಳುತ್ತೆ ಅಂತಂದ್ರೆ, ಬ್ರಯೋಫೈಟ್ಸ್ ಆರ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಆಸ್ ಆಂಫಿಬಿಯನ್ಸ್ ಆಫ್ ಪ್ಲಾಂಟ್ ಕಿಂಗ್ಡಮ್. ಸ್ಟೇಟ್ಮೆಂಟ್ ಬಿ ಏನು ಹೇಳುತ್ತೆ, ಬ್ರಯೋಫೈಟ್ಸ್ ಕ್ಯಾನ್ ಲೀವ್ ಇನ್ ಅ ಸಾಯಿಲ್ ಬಟ್ ಆರ್ ಡಿಪೆಂಡೆಂಟ್ ಆನ್ ವಾಟರ್ ಫಾರ್ ದೇರ್ ಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್. ಬ್ರಯೋಫೈಟ್ಸ್ಗಳು ಉಭಯವಾಸಿಗಳು ಅಂತ ಹೇಳಿದರೆ ಬ್ರಯೋಫೈಟ್ಗಳಿಗೆ ಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ಗೆ ನೀರಿನ ಅವಶ್ಯಕತೆ ಇದೆ ಎನ್ನುವುದು ಎರಡನೇ ಸ್ಟೇಟ್ಮೆಂಟ್. ಹಾಗಾದರೆ ಇವೆರಡು ಸ್ಟೇಟ್ಮೆಂಟ್ ಸರಿ ಇದೆಯಾ, ಒಂದು ಸರಿ ಇದ್ದು ಮತ್ತೊಂದು ತಪ್ಪಿದೆಯಾ ಅಂತ ನೋಡೋಣ. ಫಸ್ಟ್ ಆಪ್ಷನ್ ಏನಿದೆ, ಬೋತ್ ದ ಸ್ಟೇಟ್ಮೆಂಟ್ಸ್ ಎ ಅಂಡ್ ಬಿ ಆರ್ ಕರೆಕ್ಟ್. ಸೆಕೆಂಡ್ ಸ್ಟೇಟ್ಮೆಂಟ್, ದಟ್ ಈಸ್ ಬೋತ್ ದ ಸ್ಟೇಟ್ಮೆಂಟ್ ಎ ಅಂಡ್ ಬಿ ಆರ್ ರಾಂಗ್. ಸಿ, ಸ್ಟೇಟ್ಮೆಂಟ್ ಎ ಇಸ್ ಕರೆಕ್ಟ್, ಬಿ ಇಸ್ ರಾಂಗ್. ಸ್ಟೇಟ್ಮೆಂಟ್ ಎ ಇಸ್ ರಾಂಗ್, ಬಿ ಇಸ್ ಕರೆಕ್ಟ್. ಸೊ ಇವು 4 ಆಪ್ಷನ್ರಲ್ಲಿ ಯಾವ ಆಪ್ಷನ್ ಸರಿ ಇದೆ? ಹಾಗಾದರೆ ಬ್ರಯೋಫೈಟ್ಸ್ ಆರ್, ಎಸ್ ದೇ ಆರ್ ಆಂಫಿಬಿಯನ್ಸ್ ಆಫ್ ಪ್ಲಾಂಟ್ ಕಿಂಗ್ಡಮ್, ಅವು ಉಭಯವಾಸಿಗಳು ಹೌದು. ಹಾಗೆಯೇ ಬ್ರಯೋಫೈಟ್ಸ್ ಕ್ಯಾನ್ live in soil but are dependent on water, that's why they are called as amphibians. Even though they live on soil but they dependent on water for their sexual reproduction, that's why they are called as amphibians of plant kingdom. So the right option here is both the statement A and B are correct and remaining are wrong options. So next we will move for next question. In which of the following plant group male and female gametophyte do not have an independent free living existence? And the options are A, algae, B, bryophyte, C, pteridophytes and D, gymnosperms. In algae, what happens here? They don't have independent existence of gametophyte. So, next question, in which of the following plant group male and female do not have an independent free living existence? In algae, option is not a correct option, and in bryophyte, female gametophyte itself is a dominant phase. It may be female gametophyte or a male gametophyte, that is the dominant phase and independent phase, so it is also not an option. So next is pteridophyte. In pteridophyte, sporophyte is a dominant phase, but whereas this gametophyte is short-lived but independent. It is also independent and we call it as a prothallus. So that is also not an option. So the right option here is gymnosperms, where male and female gametophyte do not have an independent free living existence. So the right option here is D. In the next question, select the wrong statement. We have to select wrong statement in the following question. Options are A, algin and carrageen are products of algae. Agar is obtained from sargassum. Chlorella and spirulina are used as a food by space travelers. Floridian starch is stored food in a rhodophysae. These are the options where we have to select wrong statement. Algin and carrageen are the product of algae. Yes, brown algae, they produce algin and carrageen. So this is the right statement. So the option is wrong. Chlorella and spirulina are used by food as a space travelers used as a food because they are very rich in proteins. They used as a tablet, they comes under tablet forms. They used by the space travel. ಅಂತರಿಕ್ಷ ಗಗನಯಾತ್ರಿಗಳು ಇರ್ತಾರಲ್ಲ, ಅವರು ಬಹಳ ದಿನಗಳ ಕಾಲ ಅಲ್ಲಿ ಇರೋದಕ್ಕೆ ಆಹಾರದ ರೂಪದಲ್ಲಿ ಕ್ಲೋರೆಲ್ಲಾ ಮತ್ತು ಸ್ಪೈರುಲಿನಿನ ಟ್ಯಾಬ್ಲೆಟ್ ರೂಪದಲ್ಲಿ ಅದನ್ನು ಒಯ್ದಿರ್ತಾರೆ. ಸೊ ಇದು ಕೂಡ ತಪ್ಪು ಸ್ಟೇಟ್ಮೆಂಟ್. ಫ್ಲೋರಿಡಿಯನ್ ಸ್ಟಾರ್ಚ್ ಇಸ್ ಸ್ಟೋರ್ಡ್ ಇನ್ ರೋಡೋಫೈಸೆ. ಕೆಂಪು ಶೈವಲುಗಳಲ್ಲಿ ಫ್ಲೋರಿಡಿಯನ್ ಸ್ಟಾರ್ಚ್ ಸ್ಟೋರ್ ಆಗಿರುತ್ತೆ. ಹೌದು, ಇದು ಸರಿ. ಅದಕ್ಕಾಗಿ ಇದು ರಾಂಗ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಅಲ್ಲ. Agar and agar is obtained from sargassum. Sargassum is an example for brown algae, but agar and agar is obtained from red algae like Gelidium and Gracilaria. That's why the wrong statement here is agar is obtained from Sargassum. So, the wrong statement here is B. Which of the following option represent the life cycle of Ectocarpus and Fucus respectively? ಈ ಲೈಫ್ ಸೈಕಲ್ ಕಲಿಯುವಾಗ ವೆದರ್ ವಿ ಹ್ಯಾವ್ ಟು ಚೆಕ್ Ectocarpus and Fucus life cycle here, whether first option is diplontic, haplodiplontic, haplodiplontic or diplontic, haplontic. Haplontic means both Ectocarpus and Fucus are having a haplontic life cycle, or whether diplontic and haplontic. These are the 4 options, and in this the right option is Ectocarpus is showing haplodiplontic type of life cycle, whereas Fucus is showing diplontic type of life cycle. Here we can see haplodiplontic means they have sporophytic phase continued by gametophytic phase. Both are almost independent and they are interconnected. In diplontic, usually which of the following option represent the life cycle of Ectocarpus and Fucus respectively? Options are diplontic, haplodiplontic, haplodiplontic, diplontic, haplodiplontic, haplontic, diplontic or haplontic. And here the right option is B, that is haplodiplontic and diplontic. Why? Because here in haplodiplontic condition usually we can see in bryophytes and pteridophytes, but the example given are algae, that is Ectocarpus and Fucus, where usually the life cycle is in generally haplontic. But here Ectocarpus is haplodiplontic. They have sporophytic phase, then gametophytic phase and then sporophytic phase, whereas Fucus is diplontic, where all angiosperms are example for diplontic, where sporophyte is a dominant phase and gametophyte is just fusion, zygote is representing, that is diplontic phase. So, the correct answer here is haplodiplontic and diplontic. So, next topic, that is transport in plants. Just again we have a quick look of synapses of transport in plants. Let us start with what exactly to be transported in plants. Plant, what all things need? It needs water, it needs nutrients, it needs gaseous exchanges, it needs hormonal exchanges, plant growth regulator exchanges. For all that, water is very essential, because maximum portion of a cell is made up of water. So, concentration of water plays an important role in transport in plants. So, here in water potential we have to know these important contents, that is this symbol is called as psi, this symbol is called as psi, which in terms unit is Pascal. Water potential, this is water potential. So, water potential is mainly how it is dependent. Water potential of a pure water, if you take a pure water, its water potential is always more. Maximum water potential or kinetic energy it is having, and at standard temperature water potential of a pure water we considered it as a 0. And when for this pure water if you add solute, then the concentration or the potential of the water becomes low or negative. That is why always solute potential is negative. And when a cell kept under a hypotonic solution, the cell becomes turgid, and the pressure created inside the cell is called as pressure potential, and usually pressure potential is positive and solute potential is negative. When solute potential increases, water potential decreases. If to increase the water potential, pressure is added to atmospheric pressure, then water potential also increases, which increases pressure potential. So, water potential is mainly dependent on solute potential and pressure potential, and based on that we can follow different we can study plant water relations with so next topic is transport in plants. We just have a quick glance on this. Water is required and food is required for the plants, gaseous exchange is required, everything how that plant can manage, because they are too long plants, and even then successfully they can transport their required material from one part to the other part. And if we consider this angiosperm as it is having a root system, stem system and leaves, where they are having heavy traffic in it, even then there is no mislead here to transport the different parts. Next we will move for plant water relations, and water is very essential, and its potential, water potential to work, its kinetic energy is very important, and the relation of water potential is psi W is equal to psi S plus psi P. Psi W we can denote it for water potential, and water potential is mainly dependent on solute potential, and that solute potential is usually negative, and it is also, water potential is also dependent on pressure potential, and it is usually positive. And next based on this we are also learning diffusion in this transport system. Diffusion plays an important role, especially for gases and liquid. And then next we will move for osmosis, and we are also studying in this topic plasmolysis and imbibition. If you move for transports, whether it is a short transport or a long transport is important. Short transport means within the cell or just neighboring cell or within the intercellular spaces to outside, especially in leaves, that is called as short distance transport, where the diffusion, here we can see in diffusion water moves from its higher concentration to the lower concentration, whether it may be gas or liquid. Next, in facilitated diffusion, as because cell membrane major part is made up of lipid, and if the material or a substance or chemicals which are hydrophilic, then it is very difficult to move through that membrane which is mainly made up of lipids. So, for that they have a specialized proteins which facilitating the diffusion of the molecules. For this facilitating the molecules, they are called as porins, and especially for water molecule movement they have 8 different types of porins called as aquaporins, through which water moves from outside the cell to the inside the cell. And for facilitating this type of specialized diffusion, the special proteins are also having different nature. They may be uniport, or they may be symport, or they may be antiport. Next, these diffusions do not need any energy is spent, and they are passive, whereas another type of transport comes which is called as active transport, which needs ATP or energy to transport the molecules, and that active transport which is also called as uphill transport. In long distance transports, xylem and phloem translocating the food and water from one part to the other part, and in the long distance xylem ascent of sap takes place, and from through roots that water moves to the aerial parts, and it is always unidirectional. And then root pressure, also the pressure created in the root while absorbing the ions and water is called as a root pressure, and when excess of water is excluded in the form of liquid due to root pressure is called as guttation, and this guttation, osmosis, diffusion are not sufficient to carry out the transport for the long distances. So, transpiration is an important mechanism where through using a plant having the leaves, and those leaves are having a stomata in their epidermis for both upper epidermis and lower epidermis of monocots and dicots having stomata which are open during day and closes during night, and during daytime when they opens they evaporates water in the form of vapor, then transpiration pool is created through which the ascent of sap is formed in the xylem and water is moved. And next, food is transported with the help of vascular tissue, that is phloem. Phloem transport is from source to sink. Here the leaves are the source where the food is prepared, and usually roots are the sinks where that has to be unloaded, and that is called as vein loading and vein unloading, and that is explained through mass flow hypothesis. So, let us move for some of the questions related to transport in plants, and the first question is, transport in plants over short distances is not proceed through, whether it is a diffusion, facilitated diffusion, active transport or translocation. Diffusion is a short distance transport mechanism, whereas it is a passive type, and facilitated diffusion is also used for short distance. So, that is also not correct. Option. And active transport is also used for short distances. So, this is also not correct option, and translocation is used for long distances, but not for the short distances. So, not proceed means translocation is the right option, that it is not proceed, short distances not proceeds to translocation. Next question is, refer the given table and select the option that is correctly fills the blanks in it. And you just in the first column we can check the properties, in the second column three different types of transports, that is simple diffusion, facilitated diffusion and active transport. And here highly selective, here they have given A. Whether simple diffusion is a highly selective? No, it is not a selective, so it is no. And then facilitated transport, whether it is a selective? Yes, it is selective. Highly selective, active transport, yes, it is highly selective. Uphill transport, simple diffusion, no, yes, and C. If you go to C, uphill transport is a facilitated transport, no. Uphill transport is active transport, yes. Requires ATP for simple diffusion, no. For facilitated transport, whether ATP is required? No. Whether ATP is required for active transport? Yes, and this is also yes. So, you just check here, no, yes, no, no. yes, 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 no. No, 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 yes, no, yes, yes, no. Here A is no and B is yes, C is no, D is also no. So, the right option here is A. When a molecule moves across the membrane independent of other molecules, the process is called, whether it is a symport or whether it is an antiport or a uniport or a imbibition. When a molecule moves across the membrane independent of other molecules. If you check here, the right answer is uniport, because in symport what happens, through these specialized porins at a time two molecules are moving in the same direction, then it is called as symport. In antiport, at a time one molecule, if it is entering inside, the other molecule is coming outside, then it is opposite in direction. So, that is called as antiport, whereas uniport is independent, means it is not required any other molecule to come along with it or coming at a time out of this. So, the right answer here is uniport, this kind of membrane transport. When a plant cell is placed in a hypotonic solution, water diffuses into the cell, causing the cytoplasm to build up pressure against the wall, is called imbibitional pressure or a turgor pressure or a wall pressure, or whether it is a diffusion pressure. Means hypotonic solution. While studying plasmolysis, we are going to study three different types of solutions, that is isotonic, hypotonic and hypertonic. Hypotonic means where solvent is more and solute is less. That kind of solution is called as a hypotonic solution. When a cell is kept in such condition, and that time what happens, water or the solvent moves from outer to inside. From hypotonic solution it moves inside. So, where inside that the turgor pressure is formed. That is why, as because turgor pressure is formed, it is not a imbibitional pressure, and wall pressure is exactly opposite to turgor pressure. Against to cytoplasm, the wall pressure is formed. So, this is also not a correct answer, or not whether it is a diffusion pressure. And here, as because cell become turgid, the turgor, because of turgor pressure, the right answer here, that pressure formed is turgor pressure. The pressure that is produced by swelling of wood had been used by prehistoric man to split rocks and boulders is due to, whether it is due to imbibition, osmosis, diffusion or exosmosis. Osmosis means where solvent molecules moves from their higher concentration to the lower concentration through semi-permeable membrane. It is a specialized diffusion where water molecules or solvent moves from its higher concentration to the lower concentration. So, here it is not the correct option. Diffusion is a movement of molecules that may be in a gaseous form or liquid. Simple diffusion till equilibrium is reached from its higher concentration to the lower concentration. So, diffusion is not a correct answer. Exosmosis means when a cell kept under hypertonic solution. Hypertonic solution means where solute concentration is more and solvent concentration is less. Such solution is called as hypertonic solution. And when we kept a cell in that, the cell cytoplasm shrinks and the cell become placid. ನೀವೆಲ್ಲ ನೋಡಿರಬಹುದು, ನಾವು ಮಾವಿನಕಾಯಿಯನ್ನು ಹೆಚ್ಚಿ ಉಪ್ಪಿನಲ್ಲಿ ಇಟ್ಟಾಗ ಅದರಲ್ಲಿಂದ ನೀರು ಹೊರಗಡೆ ಬರುತ್ತೆ. ಆ ನೀರು ಮಾವಿನಕಾಯಿಂದ ಹೊರಗಡೆ ಎಲ್ಲಿಂದ ಬಂತು ಹಾಗಾದರೆ? ಬಿಕಾಸ್ ಆಫ್ ಎಕ್ಸ್ಆಸ್ಮೋಸಿಸ್. ಸೊ ಇದು ಕೂಡ ಸರಿಯಾದ ಆಪ್ಷನ್ ಅಲ್ಲ. ಸೊ ದ ರೈಟ್ ಆಪ್ಷನ್ ರಿಮೈನಿಂಗ್ ಹಿಯರ್ ಇಸ್ imbibition. Imbibition is the right option because it is also a specialized diffusion where the water is diffused through solid colloids, and this technique is used for to create a pressure. It produces tremendous pressure that the swelling wood can break the or split the rocks and boulders. Because of that only seed emerges out from that hard seed coat because of this imbibition pressure. So, the right option here is A. Next question is, the bulk movement of substance through vascular tissue of plant is called bulk movement. Bulk movement of substances through vascular tissues. Whether it is root pressure, no, the pressure created inside the root is called as root pressure. This is not the correct answer. Transpiration is evaporation of water in the form of vapor through stoma or stomata. So, this is also not the correct answer. Guttation, again the water is lost in the form of liquid through specialized structures called as hydatodes, which are present at the some of the leaves tips, that is called as guttation. So, this is also not an correct answer. And through vascular tissue, a bulk movement, mass flow of substances takes place, that is termed as translocation. So, the right option here is B, that is translocation. Next question is, opening and closing of stomata is due to the hormonal change in the guard cells, change in the turgor pressure of the guard cells, gaseous exchange, or whether it is a respiration. Is any hormonal changes takes place in the guard cells which is responsible for the stomata closing and opening? No, there is no such hormonal change takes place here. And of course, gaseous exchange takes place in through stomata, that is carbon dioxide get inside and oxygen coming out through this stomata only, but these are carbon dioxide or oxygen is not responsible for the opening and closing of the stomata. Whether respiration is responsible for the opening and closing of the stomata? No, it is also not responsible. Change in the turgor pressure of the cell is responsible for the closing and opening of the stomata, because that guard cell inner wall towards the stoma is thick and elastic, whereas outer one is thin, and when there is a turgidity inside the cell, it pulls outside the thin wall of the outer side of the stoma, pulls out and stoma opens, and when the cell become placid and it shrinks and stoma closes. Okay, stoma opens and closes due to the change in the turgor pressure of the guard cell. So, the right option here is B. Which of the following criteria does not pertain to facilitated transport? Whether it that is requirement of a special membrane protein, highly selective, required ATP energy, or transport saturates. C, in facilitated transport, that is facilitated diffusion, short distance transport, there we have learned that it requires specialized membrane proteins which allow that hydrophilic substances to move through that plasma membrane. So, this is correct option. Yes, it requires special membrane proteins, and then yes, it is highly selective. Those proteins are highly selective in nature and they transport at its saturations. All the special protein membrane proteins actively participated and its highest at transport at this stage when saturates. Transport saturation is also correct option, is also correct option, but here they want does not pertain to facilitated transport. Yes, here all these three are required, but ATP is not required. It is required in active transport and not in facilitated transport. So, the right option here is it is not required. So, the right option here is this, as because this is not required, not ATP is not required, but here the option given is required. So, this is the right option. In which of the following manner in which of the following manner does cellulose membrane microfibril oriented in the cell wall of the guard cells to facilitate opening of stoma? You just see that guard cell is having cellulosic microfibrils. How they are oriented and how they are helpful? They are helpful in opening and closing of the stomata due to their orientation. How they are oriented? Whether they are oriented, option A, horizontally, whether they are horizontally or they are randomly microfibrils or longitudinally or radially. So, you remember in your textbook diagrams how it is arranged, you just check it, dear students, how they are radially arranged. This radial arrangement of microfibrils helps that facilitate stoma to open and close. So, the right option here is radially, it is not longitudinally, randomly or horizontally. So, the right option here is D. Which of the following pathways represent movement of water from one cell to another cell through plasmodesmata? C. Apoplast pathway, symplast pathway, vacuolar pathway or respiratory pathway. So, apoplast pathway is usually non-cellular component of the tissue, that is usually intercellular space or the cell wall movement is called as apoplastic pathway. So, this is not the correct answer. Vacuolar pathway is not a correct answer, and here it is not related to any respiration. So, it is not a respiratory pathway, and the right option here is symplast pathway, through living component, cell to cell, through the connection between two cells there is a plasmodesmatal connections are there through which that transport takes place, and this kind of transport is called as symplast method pathway through which the molecules moves, and so the right option here is symplast pathway. B is the correct option. The transpiration driven ascent of xylem sap does not depend on the following physical properties of water. Then what are the properties of water? Here these are the options, that is cohesion, adhesion, surface tension and guttation, and the question is does not depend. We have to check does not depend. You just check the physical properties of the water first. Yes, they have a cohesive properties. Two water molecules, they are attracting each other. Between the two water molecules there is a cohesive force, and then they have adhesive force between adhesion between water molecule and xylem vessels. Within that lignin deposition is there, different type of forms of lignin deposition is there in the xylem. Through they have affinity towards water molecule, then there is a attraction between water molecule and xylem vessel, that is yes, that water is having adhesive property, and then they create a surface tension. When they are in liquid form, they have capillary power and they show surface tension, and then the next option is guttation, does not depend, means this water property nowhere concerned with this guttation. All these three are the properties of water. So, does not means this is the right answer, and these three are the properties of water. So, the right option here is guttation. Girdling experiment is demonstrated to prove which of the following tissue is responsible for translocation of food. Girdling experiment means if stem bark is removed in the stem where that phloem is present, and after few days if you check, you will find the swelling here, not here, not from the downward, it from outward, from upside to down there is a swelling, means this shows that which vascular tissue is responsible for the translocation of food. And you just see here xylem is not translocating the food, not the stomata or not the roots, and the translocation of food is through vascular tissue. Phloem is proved with this simple girdling experiment. So, the next question here is, transpiration in plants is not affected by external factors like temperature, light, humidity, chloroplast. The question is external factor. Yes, temperature plays an important role in transpiration. Light is an external factor which plays an important role for, and humidity also plays an important role in transpiration, and chloroplast is an internal factor, it is not an external factor because it is present within the leaf. So, the correct option for this is answer D, that is chloroplast. What percentage of water reaching the leaves is used in photosynthesis and plant growth? The water which is absorbed through roots through xylem, how much amount is used up in photosynthesis, whether it is whatever that absorbed, all the 100% is used, or more than 10% is used, or less than 1% is used, or 20% is used. It is very interesting that just less than 1% is used in photosynthesis and for plant growth. So, next we will move for chapter 15, that is plant growth and development. Again we have a quick glance for plant growth and development. Growth in plant is indeterminate and it is also measurable. It is having different phases in growth, and growth can be measurable like arithmetic and geometrical. In arithmetic, in both the dividing daughter cells, one retain its division capacity, another start maturing, whereas in geometric both the cells are having that divisional properties, that here we can say 1 plus 2 plus 3 plus 4, and here 2 square, 2 cube, 2 fourth, like that in geometrical phase we can study. And here usually in arithmetic it is linear, whereas in geometric we can find initially it is very slow and then it reaches its maximum and it saturates. That we can see here, that giving a root elongation example with arithmetic, this linear graph, and whereas this sigmoid, peculiar sigmoid, is for geometrical growth. There are different conditions are also required for growth, external conditions and internal conditions, both are very essential for the growth. And then differentiation of the cell takes place after cell division. Cell matures and differentiated to perform particular function is called as differentiation. De-differentiation, once mature cell performing a particular functions again regain the capacity of division and that cell becomes de-differentiated, and then re-differentiated means the de-differentiated cell again de-differentiated to perform a particular function. And then there are development of the growth takes place usually from seed to senescence. And then there are plant growth regulators which are very essential in growth of the plant. They are auxins, cytokinins, gibberellins, abscisic acid and ethylene. And plants are having their clock, that is based on its light for flowering, and that is photoperiodism. And then this topic also includes vernalization, where chilling effect or cold treatment is given for the flowers. So, let us move fastly for the first question, that is increased vacuolation, cell enlargement and new cell wall deposition are the characteristics of which phase of the cell growth, whether it is a meristematic phase or elongation phase or maturation phase or the differentiation phase. That is increased vacuolation, cell enlargement and new cell wall deposition is not taking place in meristematic because they are continuously dividing cell. So, this is not the correct option, and not during maturation where complete growth is happened here at this stage in the cell. So, not this differentiated means they are already performing particular function. So, this phase is elongation phase. Next question is, the living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain condition. This phenomenon is termed as whether differentiation or de-differentiation, redifferentiation or plasticity. Okay, differentiation means matured cells already differentiated is not a answer for here, and redifferentiated is not answer because once the cell is de-differentiated from the differentiated cell, de-differentiate to regain the cell division is a de-differentiation, and de-differentiated cell again becomes mature to perform a particular function is called de-differentiation. So, this is also not a option. Plasticity is changing its structures according to environmental needs is a plasticity. Heterophilia is a good example. So, plasticity is also not a correct option. The right option here is de-differentiation. Plasticity is the ability of whether a plant responses to environment or phases of life to form a different kinds of structure. Option B is cell to lose the capacity to divide, C cell to regain the capacity to divide, D is a maturation. Which one is the plasticity? Cell to lose the capacity to divide is not an option, that is become differentiation. Regain capacity is de-differentiation, and maturation again de-differentiation. Here plasticity means at the young age, at the juvenile age, they have a different structure. If you take an example of heterophilia, which is a very good example for plasticity, at young juvenile phase leaf is having different stage, but where at maturity the leaf shape changes. So, at young stage and at the adult stage it varies. A very good example, buttercup, coriander and cotton plant, we can give example for this kind of plasticity due to environmental needs and requirements, that plasticity takes place in those plants, and that is termed as plasticity. So, the correct option here is A. Which one of the following plant growth regulator promotes nutrient mobilization that helps in the delay of leaf senescence? Whether it is a ethophon or 2,4-D or cytokinin or NAA, and the right answer here is cytokinin. Cytokinin help that leaf to delay its senescence, means senescence is delayed, postponing by the effect of cytokinin. It is not through ethophon, which is commercially available ethylene, not from the 2,4-dichlorophenoxyacetic acid is a auxin, not through this, or not through naphthalene acetic acid or auxin, not through this. It is through cytokinin, where nutrient mobilization that helps in the delay of leaf senescence. So, the right option here is C. Name the plant on which Charles Darwin and his son Francis Darwin conducted experiment to prove that coleoptiles, when exposed to unilateral light, they responded by growing towards light source. And the options, they worked on which plant? The options are jawar, wheat, rice and canary grass. Wheat, they have not worked on wheat, they have not worked on jawar, they have not worked on rice, they have worked on canary grass to study in the root apex there is a presence of auxin, which shows phototropism, means they grow towards, if here it is auxin and light is supplied from this direction, this bend towards the light. This is they have observed in canary grass. So, the right option here is C. How does decapitation helps in tea plantation? Decapitation or removal of apical bud. how it helps for tea plantation. It releases wound hormones, results in the growth of lateral buds, apical shoot grows faster, internodal elongation takes place. How decapitation helps? It is not releasing any wound hormone, so this is not the right option, as because auxin is present at the apical tip, and if you remove, then apical should grow faster. No, it becomes slow. Here we are removing apical dominancy is removed due to presence of auxin, so this is also not a correct option. Internodal elongation takes place. Internodal elongation, yes, takes place if you apply gibberellic acids, but no, here we are removing apex of that stem, so internodal elongation also does not takes place. So it results in the growth of lateral buds is the right option. Here that apical dominancy, where that apical bud is removed, and due to removal, here we can see lateral buds are growing and maximum branches are formed in tea plantation. So the right answer here is results in growth of lateral buds. Gibberellins promotes bolting in whether in apple or in rose or in cabbage or in tobacco. Apple gibberellins are used to give the shape, particular shape and size to the fruits, apple fruit size. In rose, gibberellin will not promote any bolting, and in tobacco also it is not promoting. Then what is that bolting? Bolting in rosette shape of plants like cabbage, where they are promoting internodal elongation of the florets, that is in inflorescence the internodal elongation takes place, and that is termed as a bolting, and the right option here is cabbage. It is not a apple, rose or tobacco, it promotes bolting in cabbage. Cabbage is the right option. Which one of the following plant hormone act as an antagonistic to gibberellins? Whether it is abscisic acid or indolacetic acid or naphthalenic acetic acid or indolbutyric acid. Which of the following is antagonist to gibberellins? And the right answer here is abscisic acid is antagonistic to gibberellins, because it breaks dormancy, gibberellins breaks dormancy and it promotes dormancy in the seeds. That is why the correct option is abscisic acid, and remaining 3 are auxins, which are not an example for antagonistic to gibberellins. The right option here is A, that is abscisic acid. Which plant growth regulator stimulates the closure of stomata and is known as a stress hormone? Whether it is IBA, IAA, GA1 or abscisic acid. Means while in humid condition, that to check the loss of water, that stomata has to be closed to store to prevent the loss of that water. They have to close during day also. That time abscisic acid helps to close those stomata, and that is why it is commonly called as stress hormone. It is not a IBA or IAA or a GA1, that is gibberellic acid. The right option is abscisic acid. The correct option is D here. Plant growth regulator present in coconut milk is identified as, in coconut milk a naturally available plant hormone is present. Which one is that is the question, and here whether it is a kinetin or a zeatin, ethylene or gibberellic acid, and the correct option which plant growth present in coconut milk is zeatin. It is not a kinetin or a ethylene, it is a gaseous hormone and not it is a gibberellic acid or a kinetin. Match the hormones given in column A with their functions given in column B. Select the correct option from the option given below. Option column A is with the hormones, column B with its related function. Abscisic acid is helpful in seed dormancy. So, this is A. Ethylene helps in ripening of fruits. So, this is B. Auxin shows apical dominancy due to presence, apical dominancy is due to presence of auxin. Cytokinin helps in cell division. Gibberellic acid promotes bolting in cabbage, just we have studied. So, this is E. So, the right option here you just see A is fourth, B is third, C is second, D is fifth, D is 2, next question, match the hormones given in column A with their functions given in column B. Select the correct answer from the option given below. Column A is with the hormones, that is plant growth regulators, and column B is with its functions. ABA, what it does in the following group B? ABA helps in seed dormancy. So, A, ethylene helps in ripening of fruits. So, this is B. Auxin helps in apical dominancy. So, it is C. Cytokinin helps in cell division, that is D. Gibberellic acid helps in bolting, that is E. So, the right option in these 4 is option C. Name the plant growth regulator which is used to initiate flowering and for synchronizing fruit set in pineapple, whether it is ethylene, abscisic acid or indolactic acid or gibberellic acid. And here the right option is ethylene. Ethylene is a hormone used to initiate the flowering and to synchronize the fruit set in the pineapple. The promotion of early flowering in plants on exposure to a period of low temperature is called, whether it is a photoperiodism or whether it is a phototropism, hydrotropism or vernalization. The promotion of early flowering in the plants is exposed to period of low temperature, that is giving chilling effect. Naturally also it in nature also it gives chilling effect to the plants to produce flowers, and that chilling effect is not a photoperiodism. Photo is related to light, so it is not that. Phototropism is also not a correct answer. Hydro is related to water, and this is also not a correct answer, and the right answer here is vernalization. So, the right option here is D. Which one of the following plant growth regulator is used by gardener to prepare weed free lawn? Whether it is, see all 4 options they have given here is auxins. In these 4 auxins, which one is used as weedicide? And gardener to prepare a free lawn. Lawn is means grass, that is monocotyledonous lawn is there, and farmer don't want any, gardener don't want any dicot weeds to be present in that, and this 2,4-D is very effective weedicide used by gardeners to remove dicotyledonous weeds. So, the right option here is 2,4-D, and it is not a NAA or IBA or IAA. So, the right option here is C. When there is no correlation between exposure to light duration and induction of flowering response, such plants are called long day plants, short day plants, vernalization or day neutral plants. Then what is this flowering response? Phototropic plants means some plants need more light to flower, some plant need a low light intensity. Then critical intensity, if it needs less than that, then they are called as short day plants, and if they need more than critical light, then they are called as long day plants. and the plants which don't need any such duration of light, those plants are called as day neutral plants. So, the right option here is day neutral plants. Pick the odd one out. Here there are 4 different hormones, plant growth regulators are given. They are auxins, gibberellins, cytokinins and abscisic acid, and we have to pick the odd one. Auxin, gibberellin, cytokinins, they are growth promoting hormones, and whereas abscisic acid is growth inhibiting. That is why the right option, odd one out, is abscisic acid. These are the growth promoting hormones. So, the right option is abscisic acid. Which among the following plant growth regulator causes foolish seedling diseases in the rice, whether it is auxin or gibberellins or cytokinins or ethylene. Gibberella fujikuroi fungi, from where that they have seen that due to infection of that fungi there is a elongation in the internode, and that disease is called as Bakken disease, and the answer for this is gibberellins. It is not auxin, cytokinins or ethylene. Bakken disease or foolish seedling disease in rice is caused by gibberellins. ಧನ್ಯವಾದಗಳು ವಿದ್ಯಾರ್ಥಿಗಳೇ. ಈ ಮೂರು ಪಾಠಗಳು ನಿಮಗೆ ನೀವು ತಯಾರಾಗುವ ಸ್ಪರ್ಧಾತ್ಮಕ ಪರೀಕ್ಷೆಗಳಿಗೆ ಸಹಾಯಕವಾಗುತ್ತವೆ ಎಂದು ಅಂದ್ಕೊಳ್ತೀನಿ. ನೀವು ಯಾವುದೇ ಟಾಪಿಕನ್ನು ಓದುವಾಗ, ಈ ಸ್ಪರ್ಧಾತ್ಮಕ ಪರೀಕ್ಷೆಗಳಿಗೆ ಓದುವಾಗ ನೀವು ಅವುಗಳನ್ನು ಫ್ಲೋ ಚಾರ್ಟ್ ರೂಪಗಳಲ್ಲಾಗಲಿ ಅಥವಾ ಮೈಂಡ್ ಮ್ಯಾಪ್ ರೂಪದಲ್ಲಾಗಲಿ ಅಥವಾ ನಿಮೋನಿಕ್ಸ್ ರೂಪದಲ್ಲಾಗಲಿ ಅವುಗಳನ್ನು ಶಾರ್ಟಾಗಿ ರೆಫರ್ ಮಾಡಲಿಕ್ಕೆ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ಈ ಕಾರ್ಯಕ್ರಮದ ಸದುಪಯೋಗವನ್ನು ಪಡೆದುಕೊಳ್ತೀರಿ ಅಂತ ತಿಳ್ಕೊತಾ ಈ ಕಾರ್ಯಕ್ರಮವನ್ನು ಮುಗಿಸ್ತೇನೆ. ಧನ್ಯವಾದಗಳು.